Hello. Uh, we'll talk about these two topics in the coming lectures, the chromatin structure and transcription and translation. This figure shows the process of transcription on the DNA. So you can see the green arrow is the DNA unpacked in the form of a structure called beads on a string. And you find which appear like leaf shaped, the red arrows, is a lot of messenger RNA have been transcribed. The first indication that there is a molecule of life or DNA came from the experiment of Griffith's transformation experiment. Griffith, in the first setup, he used an S strain of pneumonia and injected the mice. This led to the killing of the mice. When he injected another rough strain, R strain, which is harmful, the mice didn't die. When he took the S strain and heat killed the S strain, inject the mouse still alive. When he combined heat-killed S cells mixed with living R cells and injected the mouse have died. So mice injected with heat-killed S cells mixed together with the live R cells, live S cells could be detected in the blood of dead mice. That means that there is a transformation of a material, of a molecule that transformed from the living R cells, took in DNA from dead S cells and transforming the R cells into S cells. We'll go fast on the structure of the nucleic acid, starting with DNA and RNA. Both are linear polymer of nucleotides, they contain the formation of the amino acid sequence and hence the structure function of protein. They have the critical components to select and align amino acids into the correct orders. They catalyze fundamental chemical reactions in cells, regulate the expression of genes. These nucleic acids function as the brain and central nervous system of the cell, while protein carry out the information and function they specify. The DNA is a very long linear molecule, contain the information, it's extraordinarily stable under terrestrial conditions. The error rate of copying of DNA is around 1 to 2.5 times 10 to the power 8 nucleotide per generation. The replication of these DNA ensure the continuity from generation to generation. If you look to RNA, there are three types, either messenger RNA or transfer RNA or ribosomal RNA, and of course you know the function of each of them. If we go through over the four main basic molecular genetic processes that happen inside the cell, you will see that the first one is the process of transcription, while that generate messenger RNA. This messenger RNA had to be spliced, RNA processing, to generate a mature DNA that migrate to the cytoplasm to be translated. The ribosome came from the transcription of ribonuclear protein when bound with the ribosomal subunit to make a functional ribosomes. The fourth process is the replication, which results to the duplication of the DNA material. So these are the four basic molecular genetic processes. If you go closer into the structure of nucleic acid, of course you know that it consists of nucleotides A, T, C, G, and U in the RNA. Nucleotides are consisting of a base, a pentose sugar, and a phosphate group. Binds to the pentose sugar by a phosphoester bond. The pentose and the base form the nucleoside. The sugar either ribose or a deoxyribose with a deoxygenated terminal on the 2' end. There are 5 types of nucleotides. 2 of them are purines and 3 of them are pyrimidines. You don't need to study the chemical structure of these nucleotides. If you look closer into the structure, you find that it consists of a phosphate sugar backbone, and these phosphate sugar backbones, each sugar is bound to the base. If you look on the chemical structure, you'll see the sugar and the phosphate diester bond. To make it simple, if you look closer to the chemical structure, you'll see that you have 3 ends, 2 ends, 3' ends, and 5' ends. For a simpler structure, stick diagram shows the location of the bases, but nowadays scientists prefer to write it in this format with the 5 end prime up in the front, and they omit the chemical structure. This is the simplest representation, and always the sequence is written from 5' to 3'. The structure of DNA, of course, have been discovered by Watson and Crick, although a lot of data have been generated previously with Rosalind Franklin and Maurice Wilkinson. The chemical structure, according to Shargaf studies, revealed that the percentage of A equals to percentage of T, as well as percentage of G is equal to percentage of C. And by the help of the high-resolution X-ray diffraction, they studied the crystal of DNA and found that in order to be stable, there are a lot of hydrogen bonds and the hydrophobic van der Waals interaction between stacked adjacent bases. And the two double strands had to be reversely oriented for the stability of DNA and to have the same diameter for the DNA molecule. The Watson and Crick base pair defines the association between a large purine and smaller pyrimidine. When you look to the helix structure, double helix structure of the DNA, you'll find that most DNA cells take the form of a right-handed helix. We refer it as a B-form DNA, and usually it's in the normal form. And of course, you can see the antiparallel double strand, the red and blue, and these orientations generate two grooves. One of them is a major groove and the other is the minor groove. These grooves are very important. They are potential hydrogen donors and acceptors. That means that these groups provide active sites for binding for other proteins that won't interact to the DNA, like RNA polymerase. The DNA binding protein can read the sequence of bases in duplex DNA by contacting atoms in either the major groove or the minor groove. If you look to the other forms of DNA, the B form, the A form, this B form, the difference between the B and A form, the A form arrives only in very low humidity, in which it's transformed from B to A. There is a large tilt of the base, and the number of nucleotide pairs per turn is 11, while the other is around 10.5. Some forms of the A form arise when RNAs bind to DNA or RNAs bind to RNA in helix, also exist in A form in vitro. This is a brief comparison between the double helix A, B, and A. You can see it's compared according to overall structure, rotation, base pairs, major groove proportion, and minor groove proportion. It's worth you to note that the interaction of any protein can bend DNA. You can see that from the figure that the double helix is flexible about its long axis. And this property allows DNA to bend when complex with a DNA binding protein, such as the transcription factor TATA box binding protein, TBP. This bending of DNA is also critical to the dense packing of DNA in the chromatin to facilitate its packing or packaging inside the nucleus. DNA can undergo reversible strand separation. The main player here is temperature. During replication transcription, the strands of double helix must be separated to allow the bases to pair with the bases of the nucleotide being polymerized into new complementary polynucleotide chain. This unwinding and separation of DNA is called denaturation or melting and can be induced experimentally by increasing the temperature of a solution of DNA. If we look closely, the strand can be separated when it's applied by heat. The DNA can the melting temperature at which DNA strand will separate is called Tm, and it depends on a number of factors. So Tm is the melting temperature at which DNA strands. It depends on the proportion of GC. More GC percentage will more GC percentage will increase the Tm. So you need to modify this. It will increase the Tm, not decrease the Tm. The ion concentration, the less positive ions, the less Tm you need. When positive ion is low, there is shielding decreased, increasing the repulsion force between the strands. Since the total charge on the strands is a negative charge, so when ion concentration become low of the positive ions, so because the positive ions do not neutralize the negative charges on the double strand, so that's why the shielding is decreased. You can use as well agents that destabilize hydrogen bonds like formamide and urea. That's mean if practically you got a TM that's above 100 degrees Celsius, it's not practical, so we need some agents like formamide and urea to facilitate the melting of the DNA. The extremes of pH also denature DNA at low temperature. Basically, both of them work the same at very extreme low pH or high extreme pH. Either the bases become highly protonated, so they repel each other, or become highly negatively charged, so they again repel each other. So we can monitor this melting or separation of DNA by UV at 260 nanometer. These bases in duplex DNA absorb less, the duplex DNA absorb less UV than the unstacked base in a unstacked base is in a single-stranded DNA. As regions of DNA strand become unpaired, the absorption of these regions increase almost twofold, and you can see from the figure, this figure, as long as we increase the temperature on a double-stranded DNA, the absorption increase, and for the single strand, the major change is not that dramatic. So we can use this principle to calculate the TM of a DNA molecule. For example, if we have this sequence of nucleotides and we want to calculate how much temperature I need to separate this sequence of a DNA molecule, so we need to calculate the percentage of GC. So you can see the percentage of GC, if you calculate it, it's around 50%. That means you count the GC, you will find 5 GCs over 10 total nucleotides, so it comes 50%. And from the figure you can find it's around 91 degrees centigrade. As we said before, the DNA can undergo reversible strand separation. So the renaturation, actually you are reversing the factor used while you were doing melting for the DNA, like lowering temperature, increasing ion concentration, and neutralizing pH. This leads to reassociation of the double helix. And of course, DNA strands that is not complementary, they will remain as a random coil, as a single strand random coil, and will not renature. Denaturation and renaturation can be used to study relatedness of two DNA samples, and they can be used as to isolate DNA molecule in a mixture containing numerous different DNA sequences. This is of course based on the complementary sequences. The torsional stress in DNA is relieved by enzymes. Many bacterial and viral DNA are double-stranded circular DNA, okay, circular molecules. These circular molecules occur as well in mitochondria and chloroplasts. During DNA replication and transcription of circular or eukaryotic linear DNA, DNA twists back on itself like a twisted rubber, forming supercoils. This induces torsional stress like a knot into the remaining portion of the molecule because the end of strands are not free to rotate. All cells, however, contain enzymes to relieve this torsional stress. These enzymes, major two enzymes, the topoisomerase 1 and topoisomerase 2. Basically, when there is like a torsional stress or supercoil, so topoisomerase 1 just sit on this stressed DNA or coil DNA and cut single strand DNA, relieve this torsional stress, and then bind this single strand again. The same job is done by topoisomerase 2, but the only difference that topoisomerase 2 cut the two strands of the DNA, relieve the stress, and then join back the two strands. So you can see here the difference between a supercoiled DNA and a circular DNA and the relaxed circular DNA that's been nicked. And from the GIF you can see how the topoisomerase 1 sits on the DNA, cuts one strand, and then relieve the stress and joins again the strand. Thank you.